Yes.
Hello.
Okay.
Yeah.
I'll
So,
try.
Dr. Sahu is famously called as the compliment man. Dr. Sahu, am I right?
You can say that.
And he has worked, consistently, we have seen him work for 20, 25 years that he has been at NCCS. But just to give a short information and his biodata, he obtained his PhD from the University of Delhi, and his postdoctoral training has been in the complement biology. And it is really commendable that he has pursued this interest right from his PhD days. And after doing his postdoc with John Lambris, he came back to India and he got the Wellcome Trust Overseas Research Fellow. He was one of the first Wellcome Trust Fellows at NCCS, I may say so. He moved to Institute, NCCS. And since then, he has pursued an interest in complement, but focused it more towards virus complement interactions. He has got many awards and many credit to many wonderful papers he has got. And currently he holds the position of a senior scientist, and he's also a JC Bose National Fellow. And today he's going to talk to us about complement. Like Dr. Kaveri already told us about the century-old story, and complement plays an important role in the story by the Nobel Prize in 1919 by Julius Burdet. And all that it was called as it is one of the factors in blood serum which works with antibodies. Though it was just known to be complementing, we know the complement area has made a niche for itself. And today, Dr. Arvind Sahu is going to talk to us much in details about what the complement system is all about. Dr. Sahu, please.
Thank you, Dr. Padma Shastri. For me, it was a pleasure. I had all great colleagues. We miss you very much here.
Thank you so much.
Soon I'll be out like you.
No, you will not be missed.
So let's see. Let me try and share my screen. Is it visible?
No, not yet.
Not yet.
Okay.
I think you have to press the green button, share screen.
Green button, share screen.
Yeah, I tried that.
Arvind, you open your PowerPoint already. Open it.
It's open.
It's open.
Now
try that.
Yeah, it's open. Okay, I think I'll ask my son to do it for me. So share karna. Here we go.
Screen PPT, no?
Yeah, this is open. This is a PPT.
Screen करने से नहीं होगा, तुम्हें notes देखने हैं.
No. So how do I share this?
Just click that actually. Click that share.
ये हो रहा कि नहीं हो रहा?
Yeah, you're
Okay.
there.
Okay,
here we are. Sure.
Okay, great. Yeah. So it's good. Anish is around, my son, so he can help me. I'm not good at it. Okay. So what I'll try to, am I audible?
Yep. Yeah, you are. Sir, please go ahead.
Okay. So what I'm going to try to do is give you an overview of the complement system and just talk a little bit about, gives just examples, its role in physiology and pathology, rather than going too much detail into how it works, what are the pathways involved, because that's going to take much longer time. But I thought of just giving an overview, which I think is sufficient. If anybody has any question, I can elaborate more on that. So, this about the complement system, the discovery, they started in late 1800s. Scientists like Fodor, Nuttall, Buchner, they all saw that if you take fresh serum, then this fresh serum has antimicrobial activity. Okay. And all we knew is that the fresh serum has antimicrobial activity. What we did not know is why this activity is there. What are the components really involved in this activity? So it was the Jules Bordet, or I think I'm not saying it, pronouncing it correctly. Srini can help me. I think it's Bordet, right?
Jules Bordet. The T and
okay.
the S are not pronounced. Jules
Okay.
Bordet.
Okay, Jules Bordet. So Jules Bordet was a person who actually looked more carefully into this phenomena. And what he found out that this is because of two components in the serum. One is the heat stable component and another is the heat labile component. And the one which is the heat stable component is the antibody, and one which is the heat labile component is the complements. And at that time, what he thought that it is a single component. And these two together, when you put it together, then you get this activity. And he also developed the complement fixation test, which helped in diagnosing many diseases, viral diseases, as well as other diseases. And later on, other people in Bordet, along with other, looked into this and said, this is not merely activity, it is a substance which can get attached. And that's how he discovered the complement fixation test. And like Srini said, and others, that he got Nobel Prize in actually 1990, he was awarded, and he received in 1920. For 1 year, for some reason, he did not go to actually receive, and he said, I am quote unquote busy doing research in the States at that time. But what he did is, what he called complement is not actually the complement. He termed this thing as alexin. And complement was a term which was given by Paul Ehrlich, who described the humoral immunity to us. So the original term, what he used was alexin and not the complement. Complement was given because it was thought that it helps the antibodies, it complements the antibody, and therefore hence the term complement. So how does complement really recognizes and marks the pathogen? Let's look at it more in detail. So let's say if you have a pathogen, let's say a virus enters our body, then what happens that this is recognized by various molecules. You know, one of them originally was described was the antibody. But the antigen antibody complex, or antibody recognizing a pathogen. And this complex, can we mute that mic, please? Okay. Can we please mute that microphone? I guess not possible. Okay, I'll go on. So these pathogens are recognized by the antibody that leads to activation of the classical pathway. But later we know that the other molecules like CRP or SINR1 or C1q can also recognize pathogens. It can also be recognized by lectins, which later on found out and termed as lectin pathway. And sometimes even if there is no recognition molecule, and this is very interesting phenomena, I will not go in detail, where the pathway is activated without the any recognition molecule, and this is called alternative pathway, and this is done by takeover process. So once the complement activation happens, the C3 convertase is getting formed on the pathogen, which then cleaves the C3 molecule, which is the central molecule of the complement. And this C3 once get cleaved into two fragments, bigger is C3b and then the smaller is C3a, which is an aflatoxin. The C3b gets deposited on this pathogen. And this deposition is actually covalent attachment of the C3b onto this pathogen surface. So once any surface is marked by C3b, then the complement system considers that as a non-self, and the whole pathway, then terminal pathway, gets activated. So once you have few molecules attached by any of the pathway, the alternative pathway loop gets activated, the deposition of many more molecules of C3b. And then the other pathways the terminal components come in here, and that's called terminal pathway C5b, C5, C6, C7, C8, and C9. And what happens is there is a formation of a membrane attack complex, or the MAC, which then results in the lysis of the pathogen, in this particular case, virolysis. So the question is then, when the complement activation happens, what really happens to C3 molecule? What we knew is that there is a large conformational change that occurs. What we did not know is how the structure looks. So Pete Gross's lab solved this problem and published in 2006 that, and he called this particular structure as a puppeteer with a puppet. So here is the entire, let's say this is the head of the puppeteer. These are the hands, this is the body. And here is what you see is the puppet. So when the activation happens, actually, there is a large conformational change, and what it is doing is a puppeteer is throwing the puppet onto the ground. And this little thing red, which you see here, is the thiester bond and gets covalently linked. And therefore, once C3b attached, it is attached covalently. So that is something I think the student should keep in mind. So the next question always what we have in immunology about the self and the non-self discrimination. So how does the complement system discriminates between the self and the non-self? Okay. So here is on the left, what you see is the foreign bodies or the microbial cells or a pathogen, which is sensed by the danger sensing molecules, which can also activate the complement system. And this can include antibody or the other molecules, lectins, SINR1 and other. And this, once activation happens, what we have is the C3b deposition, and that is the opsonization of the pathogen. A large number, something like 1 million molecule can deposit within 15 minutes of complement activation on the foreign pathogen. And then the terminal components come into play, and what you have is a lysis, okay? And we also have danger sensing signals, which are C3a, C5a, or what we call as anaphylatoxins. And they can kick in also the adaptive immunity. The question is then how the complement system really understands that this is our healthy cells. Is it that the C3b doesn't attach to our healthy cells? Or is it that it does attach to the healthy cells, but then it gets inactivated immediately? And the answer is the second one, that the C3b does not have the ability to discriminate between the self and the non-self. And this is something I worked during my postdoc. It can get attached very nicely onto the healthy cells also. But once it is attached, then the regulators, so our cells are lined with various regulators, and they come into play and they inactivate the C3b, which is deposited on our cells, and that's how the complement system is inactivated. But if you have a problem, if the regulators are not there or they are down-regulated, then we have a disease. So what about the regulation? And what we have is the regulation can happen at multiple steps. It can happen during the initial part of the, before C3 convertase formation. We have C3 inhibitor, right? And that can work on C1R, C1 molecule, particularly acts on C1R and C1s and inhibits their activity. Also can act on the MASP and inhibits the MASP activity, and they regulate complement. Then we have a battery of a large number of regulators which can act at the C3 convertase, and these belong to a family called regulators of complement activation, or RCA, and they inhibit the C3 convertase, which is essential for the cleavage of C3, which is the central molecule, because everything, all the effectors come after the C3 cleavage, and therefore this is very, very important step. And so we have a large redundancy is here, and multiple components can act on this, multiple regulators can, and these primarily belong to the RCA family. Then we have also C3A and C5A. These are two potent anaphylatoxins, which are there, and they need to be inactivated. And we have carboxypeptidase, which basically cleaves the terminal arginine, which is essential for the binding of these anaphylatoxins to the receptors. And that is how, so the carboxypeptidase removes that arginine. And we have CD59, which blocks the MAC formation. Basically, it binds very tightly to C5b through 8 or even C5b through 1 or 2 molecules of C9, and blocks the C9 interaction and polymerization. And that's how the MAC is blocked. So we have regulation in place at multiple places. So what about the central inhibition that is done by the RCA, or regulators of complement activation? How do they regulate the C3 convertase? So here is, they regulate by two different activities. One is called the decay acceleration activity, and another is called the cofactor activity. So what it essentially means is that once the C3 convertase is there, which is nothing but the C3b with, let's say, a BB, which is a serine protease here, part of the convertase, or C4b and C2a, which is again has a serine protease part. This is an enzyme. The RCA molecules bind to that, okay? And once they are bound, they dissociate, okay? They accelerate the dissociation of these particular BB or C2A. And because they help in accelerating the decay, this activity is called decay accelerating activity or decay acceleration activity. Now, another thing they also do is that because the C3b, if it is there, can form the convertase again, which must not happen. So what they also do is that once they are bound, they recruit a protease called factor I. And this factor I, once is recruited, can inactivate the C3b into inactivated form of C3b, and therefore this inactivated form of C3b can no longer form the C3 convertase. And because it acts as a cofactor, this activity is called a cofactor activity. So we looked into this because in our lab into more detail, and what we found is that during this activity, what it does is that the C3b molecule, this is very flexible. So this particular part, so this is the one which is attached to the surface here. And this part is very flexible. So what the regulators do is that they actually breach this particular region. Okay, let's not go in detail into the domains, but it basically bridges. And once it's bridged, then the serine protease can come and cleave here. This is a portion, this is called cup domain, where it is cleaved. And this is how the inactivation happens. We also look into the decay into more detail, and what happens is that once this regulator is bound, actually it interacts with the VWA, and it induces a conformational change. Actually, this is an allosteric change. I'll not go detail into the mechanism, but then this is how it generates allosteric changes, and which is much away from the actual binding site. And then this is how it is decayed. So let's look into the role of complement in the physiology. What does it really do? So what we knew earlier is that, and what Srini also talked about is that, it helps opsonization. And once opsonization is there, obviously the C3b molecules are opsonized, and that will help the phagocytosis through the complement receptors. Okay. And complement receptors are present on various immune cells: neutrophils, monocytes, mast cells, macrophages, dendritic cells, B cells, and T cells. Meaning that if the receptors are there, then the complement system must interact or have a crosstalk with these cells, which has been explained very nicely in last 3 talks, okay, that these cells, what do they do? So therefore, what I want to put in here is that the complement cells can actually interact with all these cells. So if the opsonization of the pathogen, that is going to help the phagocytosis. I talked about the MAC formation and lysis of the pathogen. So that's also, it can remove the pathogen or lyse the pathogen. We have anaphylatoxin generation, particularly C3A and C5A. These can act as a chemoattractant, and they can attract the neutrophil. For example, C5A is a very good chemoattractant for the neutrophils and also the macrophages and monocytes. Now they can also cause cellular activation, and therefore inflammation also is there kicked in because the recruitments of cells are there at the site of infection. So these are all the effects directly we knew earlier. Later on, when people looked more carefully, then we also understood that it can also act as a disposal system. So it can help in clearance of immune complexes. Okay. So here and as well as the apoptotic bodies. So complement can not only help in removing the pathogens, which is the role everyone knows, but it can also help actually in removing the immune complexes and apoptotic bodies. What we understood in last 10 years that there is also crosstalk of the complement with the B cell and the T cells. Okay. And there is an augmentation of antibody response by the complement and also promotion of the T cell response. It can also actually help in elimination of self-reactive B cells and also help in immunological memory. So there is a good amount of crosstalk of the complement with all the other cells, okay, including B and T cells. So I'll not go in detail, but this, the role of complement in antibody formation was actually first demonstrated by Pepys in 1970s, early 70s, I think 72 or 73. What he did is that, cobra venom has a component called cobra venom factor. And this is something like a C3 like molecule, and when you inject, it forms a very stable convertase, and once you inject, it can virtually deplete complement from almost any animal. And this turned out to be a very good reagent when anybody wanted to deplete the complement from any species, whether it's a mice or even works with humans, anything. So what he did is that he depleted complement in mice, and then he injected the antigen, and what he saw is that the antibody response is almost like 50%, okay, and whether they are T dependent or the T independent antigen. Later on, people looked more carefully, and it was Doug Fearon who actually demonstrated, published in Science, is that if you take any antigen, and he took hen egg lysozyme, HEL, and attached that, this is a covalent attachment of the C3 fragment called C3d. And if you do that, because the B cells have a receptor, which is CR2, and the ligand for CR2 is C3d, okay? So Srini talked about this and also, this was described earlier, okay? Dipankar also talked about B cell and antigen recognition. But what I'm showing you here is that it's not just the antigen recognition by BCR, but antigen coupled to the C3d molecule, which can be recognized by the CR2. And when this happened, the threshold of activation requirement of the antigen is very, very low. So in other words, if you take HEL and attach the C3d molecule, 2 or 3 copies of that, the requirement, if you get, let's say, X amount of antibody generation, then the antibody generation can be increased up to 100 to 1,000 fold. And therefore the C3d can act, or complement can act as a molecular adjuvant, okay? But it took almost 10-15 years for Doug Fearon to show this. We also know that the dendritic cells, follicular dendritic cells, have CR2, and that can hold the antigen. So antigen retention can also happen. And therefore complement plays a very important role in the antibody generation. So I'll not go more detail into this, but please remember that if there is no complement, then the antibody response is very poor. And this has been shown in various animals, not just mice, but mice, guinea pigs, dogs, and so on and so forth. And here is the mechanism how it works. Then what about the T cell? Dipankar nicely talked about, and also Srini talked about, cognate recognition. So there is antigen presentation, right? So we have co-stimulation that can happen. So here's the one, the antigen presentation on the MHC recognized by the T cell receptor, and for the co-stimulation CD28, CD80 and that's why the cognitive recognition, signal 1 and signal 2, as it's called, there is the T cell activation. But what also happens when people looked into this, particularly Claudia Kemper's group at the NIH, currently, she was at Cardiff earlier, that there is a complement synthesis and activation that happens. And both these cells, the antigen presenting cells as well as the T cells, have complement receptors. Okay, so C3a and C5a are produced, and these are recognized by these receptors. And when this thing happens, what you have is the generation of Th1 cells or the Th17 cells. But if complement is not there, let's say you take, receptor deficient or the complement C3 deficient, then what you will get is the generation of the Treg. So complement also plays a role in the polarization of T cells, and it decides. So this is something I'll not go into the detail into the mechanism, which has been worked out very well. So we also looked into this, and what we did is that we took the flu virus. This is that in 2009, H1N1 we had everywhere. So we took this strain from NIV and collaborated with NIV and took it and said, okay, if this is so important, what about the virus? Take the flu and decide. And we took complement C3 being a central component. We looked into that. What happens if you do the infection here, right? Let's look into not in vitro, but in vivo scenario. And what you see is that if you take C3 knockout mice, you have loss of weight significant. And if you look at the mortality, pretty much in about day 10 or day 11, all the mice die. Okay, so if you take C3 knockout mice and do a flu infection, H1N1 infection, they are highly susceptible and they die. Okay, what about then the pathway requirement of the C4 or the factor B? And we saw that they also participates both classical, and you have more death, but obviously what you see is more death in this C3 knockout mice as opposed to C4 or factor B knockout mice. And what we showed in this particular paper, that because of complement activation, there is more generation of specific antibodies against the HA, which is the surface protein of the flu virus. And that is why it protects. So I'll not go into the details of this paper, just want to show you that if the complement is not there, then yes, they are susceptible to the viral infection, particularly H1N1. And this is because you have less generation of specific antibody. And if a complement is really important, then viruses must have developed a mechanism. And this is true. There are multiple mechanisms generated by all sorts of pathogens, including viruses. And this is where our interest lies. And we are working on the pox viruses and the herpes viruses, which have pirated the protein, which are like regulators, human regulator. And that's where my lab mainly focuses on, but we'll not get into that. Now, what about the other roles of complement in the physiology, right? So this is in around, I think, 93, 94, when I was visiting Washington University. People found out that this was used as actually control the liver cells because it was expected that the hepatocytes will not have the complement receptors. It's only the immune cells were known to have complement receptor. And they looked into that, and they were surprised because the control turned out to be positive, and they said, you messed it up, but then it turned out to be true. They published that even the non-immune cells have complement receptor, although they did not know what is the significance of this. Later on, when in John Lambdis's lab, where he's my mentor, I worked, he was interested in looking into the role of this, if anything, complement plays in regeneration. And so, you take axolotl or the newt and you cut basically, a limb, the limb regrows, we know that. And what happens is that, wherever we cut, there is a dedifferentiation of these cells, and then they redifferentiate. And when they looked into that, this is called something called blastema, when the dedifferentiation of cells happen, and a lot of cells are migrating there. The blastema cells, as well as the wound epithelium, make a lot of C3. And later on, the lab moved into looking into and found that complement plays a role in even in regeneration in mice. Okay, so if you don't have complement, and particularly C3 and C5a receptor, the regeneration is perturbed. So here's another role complement can play. I'm not showing all the data. We know that the folate deficiency, if it is there, that causes neural tube defect, right? So here is you can see the graph. This is a replete food. Here is a deficient, folate deficient. And what you see here is that there is a defect neural tube. But if you actually block the complement receptor, particularly C5a with the antagonist, what you see is that there is a large amount of deformations in this and many cases. So what they have shown is that the complement receptor, if you use receptor knockout or use a receptor blocking antagonist, then the neural tube is there, meaning that complement does play a role here as well. During development, there is a collective, particularly neural development, there is a collective migration of the cells, okay? And so when people looked into that, what they found is that, yes, there is a collective. I don't know if I can play this movie. No, the media is not working. So I'm sorry for that, but what I wanted to show is that if you take C3a, then this cells basically migrate here. If you use a Desarge form, which is the inactive form, the cells don't migrate here. Or if you use a scrambled C3a peptide, then also the cells don't migrate here. So during development also, complement seems to play a role. What about the role in the pathology? Okay. So as I said that, our cells are protected because we have complement regulators. Our cells are lined by complement regulators. And so here are the examples, right? PNH, where the CD59 is not there, then we have a PNH, basically RBC lysis. Hereditary angioedema, C1 inhibitor absence causes this. We have factor H deficiency, then you have, linked to age-related macular degeneration or AMD. We also have kidney problem because factor H plays a very important role in kidneys. So we have MPGN2, or also called dense deposit disease, and atypical hemolytic uremic syndrome, or AHUS, where complement mutations are there, then we are in trouble because the factor H is unable to regulate. And those who are in this kidney business will know that AHUS is also very common in India, so Dr. Professor Arvind Bagga at All India Institute of Medical Sciences has cohort of almost 100 kids. And what he has shown is that in India, we have actually autoantibodies against factor H, and that is leading to AHUS. And we know where these antibodies bind. I had interacted with them and we worked together for some time. But there is also can be overactivation of complement, and this is linked to many. This is not just infectious disease, sepsis, anaphylaxis, ischemia, reperfusion injuries, psoriasis, myasthenia gravis, and we can go on and on. Stroke, and Srini also pointed out to this, rheumatoid arthritis. So many diseases, whenever there is a complement activation, so it does help, complement does help us to protect from the diseases, but if there is an overactivation, then that can cause a lot of injury, either directly or indirectly by recruiting cells. So this is something I did at NCCS with the help of Dr. Mohan Vani. His interest is osteoclast, and arthritis. So we know that the osteoclast and osteoblast, there's a balance. You have more osteoclast, then you have bone damage, bone resorption. So we wanted to see if the complement really plays a role in this. And what we saw is that if you use C3 knockout mice, you don't really get this giant trap positive cells which are osteoclast. You have this, and you put the C3 back, then you can reconstitute it, basically this. So during arthritis, what complement plays can be directly their effect by the inflammation, by recruiting cells, or through the MAC, but can also be due to more the osteoclast formation. Now during cancer, what has been shown, and this was initially people used to believe cancer cells, because they have a lot of regulators, there is no complement activation, therefore, and they are protected from complement. But now what we know is that, complement also actually causes inflammation. And in this particularly, cancer tumor cells, and can act. So what people have shown is that if you have, for example, tumor cells which are making more C3 or less C3, what you see is that the tumor cells which make more C3 are more aggressive in nature, because there is some kind of inflammation that is going on through generation of C3A and C5A, and but it does multiple things actually. It also helps the macrophages to turn into the anti-inflammatory type or M2 type. It can also recruit, the C5A can recruit the suppressor cells, the MDSCs here, which can also complement. If it is there, then what you have is a tumor. And this is not just the complement which is synthesized by the tumor cells, but also the host, right? So if you take tumor cells and in the artificial scenario, if you take, let's say, melanoma cells and put it into mice, which are C3 knockout, then the tumor formation is very, very retarded, very small. And Girdhari Lal, who is going to talk here probably tomorrow, he is looking into this more carefully, into the host complement and the tumor, how it occurs and how complement really promotes this. This is something in the COVID scenario, I thought, since everybody might be interested in knowing what happens during COVID or SARS-CoV-2, does it really activate complement? And the answer is yes. What happens is in this case, as opposed to what I showed you in H1N1, where complement was actually helping, and if you don't have complement, then the mice die because the virus survives. Here exactly opposite happens. There is a hyperactivation of complement. And because there is a hyperactivation of complement, then there is a large recruitment of neutrophils, macrophages, netosis happens. And because of this, the inflammation is really high, and complement activation has been shown. And there are papers in PNAS and other places you can see it, that complement. So what has been also people are doing is clinical trial into this, that what will happen if we block complement. So one, what they are using is the anti-C3 therapy and anti-C5 therapy, because if you block C3, that's a central component, then you will not have C3A and C5, and that might help. So when I was in John's lab, I identified a peptide called comstatin, and comstatin, when it binds to native C3, blocks its activation. John kept on, we had patent on that, but he kept on working on this and made much more potent analogs of comstatin. And he is trying this, and the initial trial looks very promising. The inflammation was less with the anti-C3 and or comstatin therapy. IL-6 and all the other inflammatory markers were very low. So they are doing now large trial. The another group at France, actually in Marseille, they have antibody against C5 receptor, and they published this in Nature and showed that this works pretty nicely. But now human trials are going on, and we will know whether this really works or not. These two large studies are going on. So I'll stop here, and I would like to conclude by saying that the complement can be a friend, but can also be a foe. And I would like to stop there. Thank you very much for patient listening.
Thank you, Dr. Arvind. Thank you so much for the wonderful, wonderful talk. It was nice listening to complement system as well as listening it from Arvind Sahu. That was really good. And thanks for making this complicated system so simple and easy to understand. And we hope you'll continue to work in this area for many more years to come now.
Yeah, let's hope so.
Yes,
Yeah.
Sure. Dr. Chandrasekhar, should I sum it up?
I think I just have one small questions to Dr.
Oh, sorry, I forgot to ask if somebody had questions.
Yes.
Sure.
No, actually, see, we have scenarios in lupus where the complements are on the lower side.
Right.
And at the contra, we have a rheumatoid arthritis where we have either complement is normal or higher. And the infection episodes seems to be a little higher in the lupus, vis-a-vis compared to the rheumatoid arthritis. Do you feel that the complement has a major role, which is lesser in the proportion? Or alternatively, we also do see the immunoglobulin response are proportionately higher in lupus, vis-a-vis compared to rheumatoid. How to explain this imbalance?
Yeah, so what happens in lupus actually, so lupus is also linked to complement. And the early components like C4 is linked to lupus. So what people believe, and more study is needed, but Mike Carroll's group at Harvard has looked into this. And what is happening is that the autoantibodies we have more if there is a complement problem, and they circulate more, and therefore you, as a result, you have a lupus. Okay, so complement is linked to autoimmunity. So complement deficiency is linked to particularly the early components such as C4, C2. These are linked, but more studies are required. Is how complement really helps us to remove the autoreactive B cells from the body. But what Mike Carroll has shown is that definitely there is an increase in such autoreactive B cells or autoantibodies in complement deficiency conditions. But the detailed mechanism has not been worked out. What complement also does, obviously, is removes the pathogens by direct effect, right? And therefore, what happens sometimes is that if the complement is overactivated and very little complement is left, then you can have more infections as well. So this can turn into that if there is more infection, but more problem with the complement during overactivation is the damage. So there's a lot of tissue damage that occurs, and therefore it is very, very important to block that. Steroids, when you use, actually it blocks the synthesis of complement. So the indirect effect of steroid could also be through, one of the ways it can work is through the complement. But like Srini I also believe is that, steroid is not good because it blocks everything. Rather than, more specific therapies, for example, monoclonal antibodies in this case, as Srini talked about others. And also Dipankar talked about monoclonal antibodies. We have monoclonal antibodies in complement system also. Like I said, Eric's group in Marseille is trying the C5 receptor antibodies, or there are C3 receptor, or the one which is the approved one is the C5 monoclonal antibodies, which is in clinic actually, which can be used and is being tried. Also in COVID, there are trials associated with this. So yeah, complement, when there is overactivation, I think it's important that to block. That the question is how much complement one should block. If you block everything, what about the infection? Then particularly if it is very long duration. And what has been seen at least in the experimental models, that even if you block 50 to 60% of the complement, that's good enough to block the complement related damages. And therefore one need not to be, or more specific, that if we have a pathway specific monoclonal antibodies, or for that matter, the small molecules, then, one need not block all of them. One may block something which is important, which pathway is important. And therefore, all what I'm saying is in pipelines, there are a lot of molecules which are being tried now, and they are in pipelines. But the one which are approved are against the C3, which blocks C3, or the C5, or the receptors which are being tried now. Okay.
Yeah, as I appreciate that, we have a real issue when we come into the clinical management.
Yes.
We do use a term rather than calling them as immunosuppressant as a immunomodulators. Either it tunes down or tunes it up. So because basically when it comes to the autoimmune management, we try to tune down the immune system. So I mean, this is one of the questions which we always have a fighting with the to counsel the patients, that they always say, I'm suppressed of immune system, and you are giving me an immunosuppression, I will have more infection. So we need to tell them, to explain them, say, look, you are like a diabetic where sugar is up and I give insulin to bring it down. So here what I'm doing is I'm not really suppressing the immune system. I'm trying to bring down the immune system, which is otherwise abnormally active. Correct. So, I mean, somehow the term immunosuppressions have a bad tag for us when we clinically try to interpolate the information. I think we have to try to call those as immunomodulators, but major challenge for us in a clinical scenario is how much and which pathway and where. These are the three questions. If it is easily answerable in a 1:1 basis, that is, the patient who is sitting across me would give a perfect solution. I think we need to we are looking forward for that.
Any more questions from anybody? There are no questions in the chat box. So should we wind up, Dr. Chandrasekhar?
Any comments from the all the speakers? Any concluding comments?
I think it's a very nice, series of the talk. First talk is come out with quite nicely. The first series of talk, I think, is that a lot of people are. I think how many people are registered?
It's around 200 plus.
Yeah, that's quite a good.
Pardon?
241, Doctor.
241.
Yeah.
That's very good.
That's very good. For the society, the immunology is getting popularized.
With inflammation.
With inflammation.
Yeah, I think we have had a wonderful 3-hour experience. And I think the way the session has been packed, First, we had Dr. Chandrasekhar give us a bird's eye view on the immune system organization. Then we had Dr. Deepankar come more specifically to the different cell types and disease-related immune cells. Then we had more specific attention paid to immunoglobulins, and we had their role, functions, applications. And we also had a down the memory lane, a century-old story, and with different Nobel Prizes and milestones during the whole process. And complementing all these talks was Dr. Arvind's talk. He complimented them by giving us a wonderful compliment cascade, which is so complicated. Even to tell that I got it complicated. So thank you, all the speakers. We have enjoyed all the talks, and we look forward to the session tomorrow. And if there are any announcements, and we could leave that now.
Yeah, can I say something?
Yeah, sure.
Yeah, what I would like to say is that if it is more interactive, that would probably help more rather than a monologue. Because what happens many times when we are giving any talk, we are not quite sure how much what we said is understood and what are the problems. So if some way you can devise a way to make it more interactive, I think that would be wonderful.
Yeah, the problem is when you have a large number, the platform interaction becomes a issue. Because you might have heard in between the noise. It's very difficult to contain.
Difficult to control. You have to keep them mute.
Yes. I think
very clear today. Too little is bad, whether it's a cell number or whether it's activity. And having more cells or more activity is equally bad. So if we are all having a good immune system and we are living happily, that only shows that we should be blessed, and we should be thankful that we are surviving well with our immune system, with all the components working in sync. Thank you. Thank you very much.
I think a small announcement to all the delegates. I think tomorrow the session starts at 10:30. Please log in 5 minutes earlier. And we will be having an interesting talk on the other 5 talks, especially on cytokines and various other pathways. And I will be looking forward to see you, all of you.
Thank you once again. Thanks very much, Dr. Vishwas.
Thank you very much.
Of sharing the virtual platform with you.
Thank you. Thank you very much. It's a pleasure and privilege.
Thank you, Doctor. Thank you, everybody.
Thank you. Thank all the speakers. Thank you very much.
I'd like to thank all the speakers. And wonderful talk and wonderful organization. Thank you. It came out very nicely.
Thank you.
Bye.
Bye.
Thanks, Iyengar. I think
Thank you. Thank you.
yeah, we'll conclude
Thank
the
you.
sessions. Good night.
Thank you. Good night.
Good night.